I've honestly loved science for as long as I can remember, even when I was a little girl. And I love discovering things. I love finding out how things work. Being able to do something that might benefit humankind in some small way is a great privilege, and that's just an added benefit. I feel very lucky to be able to do what we do, and to be able to do it here. It's just been fantastic. I'm really very grateful to have been able to have this kind of an adventure. Another thing about research is that it takes you to places where you never thought you would end up. That's part of the fun. I think every answer leads to a new set of questions. And so from an investigative, sort of intellectual point of view, you just don't know what the next question will be, and that's part of the adventure. We're on the cusp of, I think, a revolution in science. The human genome has been sequenced. We know pretty much all of the genes that make up human beings, all 25,000 or so. And now we have this wealth of information at our fingertips, and we just need to use it to now learn as much as we possibly can about human health and disease. We work on the cell biology of endothelial cells. These are the cells that form the lining of blood vessels. And we're interested in understanding how these cells form new blood vessels in a process called angiogenesis, and also how the endothelial cells keep the blood in the fluid state and prevent it from clotting when it shouldn't clot. Blood clots are bad for us. They can cause heart attacks and strokes. And new blood vessels can be good in some situations, but they can be very bad in other situations. If you have a tumor, blood vessels can promote the growth and spread of the tumor. Or if you have diabetes, the excess growth of blood vessels in the back of the eye can cause loss of vision or even blindness. So here in the lab, we're trying to understand exactly how the endothelial cell contributes to these processes at the level of the molecules. And so we have the ability to grow endothelial cells in culture, and we use models of human disease, and we use a variety of biochemical tools to try to understand the pathways that promote the growth of new blood vessels or tell blood vessels when not to form a clot. We'd like to understand the molecular pathways that lead to cancer angiogenesis on the one hand, the blood vessels that feed a tumor and allow it to get bigger or to spread. And we'd like to understand the pathways that allow the endothelial cell to keep the blood in the fluid state. So we want to be able to prevent blood clots, and we want to be able to prevent vascular proliferation, the increased growth of blood vessels when that should not happen and when it contributes to disease. With some of the work that we're doing, we may have identified a gene that is mutated or defective in some patients with thrombosis. We're looking into that now. And secondly, some of the work that we're doing, I think, may offer a new treatment for abnormal angiogenesis in the retina and possibly even in tumors. 5 years may be a little premature to say that we'll have a treatment for humans, but I think we will have a new approach and we'll have the basis for investigating new treatments. It's been fantastic to be able to do this and have this adventure here at Weill Cornell. It's a wonderful place to work. We have fantastic colleagues, all the way from the students who teach me every day, to our postdocs, to faculty colleagues, to our neighbors at our associated institutions, and even to our colleagues in Ithaca with whom we're also collaborating. In addition to all the great people that are here. We have state-of-the-art facilities, and most of all, it's the people.